As we have learned so far, the main use of quinoa is seed, and it is where the highest nutritional component can be found. Here is a scanning electronic microscope photo of quinoa seed section. The embryo forms a ring around the tissue where starch reserves are accumulated. This tissue is called the perisperm. The embryo is where most protein and fat reserves are accumulated. Other tissues are relevant for quinoa use. The embryo is surrounded by two tissues. The pericarp and the episperm are both known as seed coat. Saponin, a group of molecules conferring a bitter taste to quinoa, are accumulated in the pericarp.
Quinoa has a higher protein content than cereals like maize, rice, or wheat. However, these values are lower than legumes, exemplified here by beans. Quinoa oils have a high quality related to a high unsaturated fatty acid levels and a close to optimal omega-3 to omega-6 ratio. But it is in the composition of the proteins where quinoa shows its highest advantages. For example, has a higher value than cereal for lysine, an essential amino acid critical for children's brain development. And these essential amino acids have a close to ideal balance. Quinoa is also a good source of several vitamins and minerals.
Starch is made of two types of molecules, amylose and amylopectin, which its molecular structures is shown here. Quinoa has a high proportion of amylopectin, with uses in the food industry. Two types of seed can be found according to the appearance of the perisperm. They are classified as amyliceous or vitreous, when the perisperm is translucent. These differences can be linked to starch composition, and farmers give different uses to them according to this characteristic.
One of the most distinctive traits of quinoa is its variation in colors and size. However, varieties different in chemical composition have been little promoted by the food industry yet. When looking at genetic variation, genotypes are grouped for agronomic performance according to their geographic origin. Sea-level genotypes, as mentioned, are the ones adapted to temperature, high-altitude environments. All the other genetic groups from the high Andes are not well adapted outside of the tropics. Molecular variation is also geographically structured. For example, for the local populations of the northwest of Argentina, the 4 genetic groups shown in the map reflect different climates, from lower altitude humid environments in the east to high altitude dry environments in the west. These genetic groups harbor a significant amount of genetic variation, as shown here by the number of alleles by genetic groups. The altitude chart shows its changes when moving from the dry highlands in the west to the humid valleys in the east. When analyzing local genotypes from Argentina for several composition traits, for example carbohydrates, proteins, fats, dietary fiber, we found no geographic structure in variation. And we found 4 genetic groups according to their seed nutrition traits combination. This means that genotypes can be promoted for some particular traits, for example for fiber in G3 and G2, or for a more even composition like in G1.
How environment affects seed composition will be the subject of our next video.